In this video, we're going to be talking about fertilization in plants and how seeds are formed. So we know that a bee can serve as a pollinator, right? What is a pollinator? A pollinator is anything that can transfer pollen from one plant to another, or in one flower to another. Why do we care about this? Because pollen is the male sperm of a flower, the male good of life, the magic potion of life, the male gametes, right? It is needed um, for a new plant to be made, right? So this bee picks up this pollen from one flower unintentionally as it was looking for some sweet stuff in the rose. And as it picks it up, it now moves along to another rose because this one ran out of sweet stuff. So it comes here now and it gets some more nectar in here, some more sweet stuff. And in the process, it drops off this pollen that came from this rose now to this rose. So what exactly happens? So let's take a bigger look at this rose here, okay? And let's open it up to see what's really going on the inside. So now this bee dropped off this pollen. Where did this pollen now land? This pollen is the male stuff, right? So we know it is made by this part here called the anther from this rose. But when this bee brings it, it's not going to go to the anther because that's where it's made. It rather wants to go to this part here called the stigma, the sticky stigma. That's how you can remember it. It is sticky and it is the female part. So it's going to catch this pollen. So this brings us one step closer to fertilization. Now what happens next? Next is very interesting. Now we're going to take a look at just this part here because that's what's important, okay? So I made a bigger picture of it, okay? Here we have it. So you can see here, this is this part, the white part with one filament and anther, because it's going to look too complicated if I put even more. So now, let's say this is the pollen, this thing here, it landed. What happens now is this pollen will germinate. Germinate means to open up and release the nucleus, because we know the DNA is in the nucleus, right? For example, your cells, the DNA is in the nucleus. The important stuff is in the nucleus. So this pollen will land here and release the DNA, the actual important thing, okay? And it's going to travel down this thing. And this thing isn't always here, okay? So this tube is called the pollen tube. Makes sense because pollen lands on it, lands here and creates this tube. So this tube isn't always there. It is created after the pollen lands. So once the pollen lands, this tube will form and dig down all the way to one of these structures, okay? You see these six, 1, 2, 3, 4, 5, 6. These are called ovules, okay? And this little big loop here that contains all of these ovules is called the ovary. The same as in humans, right? A human has an ovary, which contains all the egg cells. So an ovary is this part here, and it contains ovules. But the ovules is the female part that's going to mix with the male part, okay? So this is the ovule. So it makes this pollen tube down towards the ovules, which has the female DNA in it. This is a cell, right? This is the cell that needs to combine with the male's DNA. Now, after the pollen tube forms, now the DNA can be released, these little black dots, okay? And they get released and they travel all the way to this ovule. And then it has to penetrate this barrier of cells. Once it does so, it will now enter the ovule and come to this little important cell here. The DNA, the male's DNA, will now combine with the female's DNA here to form a zygote. So a zygote is the cell formed, or the stage of development, when the male and the female DNA have mixed. So this will now duplicate and multiply and grow until it forms a seed, okay? So that's what happens. That's how fertilization occurs. Now, what is this little hole here called? This little hole here is called let me show you the name. It's called the micropile, okay? So this small hole here is where the pollen tube digs into, okay? And this little small hole here will be very important later on during the seed development. So now that we have the zygote, now that fertilization has occurred, this will take some time until this duplicates enough until a seed is formed, okay? So after some time, this will become the beautiful seed, okay? A seed. Seeds have many different shapes and forms. I'm just showing you a random one here, right? So, and inside the seed, there's also some important structures that we're going to look at now. Let's take a look at them. So depending on what type of seed we have, so there are two types of seeds, Dicotyledon seeds and Monocotylidon seeds. So depending which type, there is slightly different structures. So I'm going to show you both. A rose, specifically a rose, normally a rose has a dicotyledon seed, but many other fruits and stuff like what do you call that? I lost my name. What is it? Corn. Corn. Yeah, corn is is an example of a monocotylon seed. So I'm going to show you both because they're both important. So let's look at the inside of these. We cut them open. We're looking at the inside. What are some names that we need to know? Okay, first we start off with the embryo. The embryo. The embryo is going to be, remember, the zygote is what forms when the male and the female combine. The embryo is what is formed when this zygote has duplicated a lot and grown for a while. After a while of growing, we call it an embryo. Okay, so this right here is the embryo, this structure right here. And this right here, I would say this is a little bit after the embryo, or maybe we can call it the embryo, because you can see the leaves are starting to form. Anyways, this is the embryo. This is the embryo of a dicotyledon seed, and this is the embryo of a monocotylidon seed. But we break the name embryo down into two. The embryo shoot and the embryo root. It will make sense. Embryo root because that part, the embryo root will become the root of the plant. Okay, so it's at the bottom. Embryo shoot will become the stem of the plant, the part above the ground, basically, above the soil. So it makes more sense on this picture of the dicotyledon. This will become the root, and then the shoot. I mean the shoot and then the root. By the way, they have another name. So the shoot can also be called, let me show you here. By the way, so there's all the definitions that I'm talking about just in word form. So the embryo shoot can also be called the plumule, and the embryo root can be called the radicle. Easy to remember because R for root and radicle, okay? Now, what's next? Next is the micropile. I already mentioned the micropile, right? The micropile was this part here where the pollen tube grew into, okay? So, where is it? It's not so clear on the monocot seed here, but it's basically this little hole here, okay? Or on the dicot seed, it's this little hole here, right? It is important not only for the pollen tube to go into, but this is going to be the part where water is absorbed from when the seed is in the soil waiting to grow into a plant, okay? Because we know water is important. Without water, the seed can't grow. That's because water needs to be absorbed through the micropile. Now, we finally get to cotyledons. Why did I say dicotyledon seed? And why did I say monocotylidon seed? Because dicotyledon seeds have two cotyledons, whereas a monocotylidon seed has one cotyledon. Mono stands for one, di stands for two. Okay, so where's the cotyledon? Okay, it's in the monocotylidon seed, it's this white part here. So it's not that big in a monocotylidon seed, and there's only one of it. In a dicotyledon, it's this big part here, this big white part here. But it has two. So obviously we cut the seed open here so we can only see one. But in a dicotyledon seed, there's two of these. Whereas in a monocotylidon seed, there's only one of them, okay? What does the cotyledon do? The cotyledon will develop into leaves later. It has nutrients that allows the seed to develop and grow leaves, okay? So that's important. Now, testa is this part here, okay? So all of these, a monocot and dicot have in common so far, except for cotyledons. The one monocot has one, dicot has two. A testa is the protective covering of the seed. It's the outer part, the most outer part. This is important to protect the seed from harsh weathers, harsh conditions, such as the seed blowing around everywhere. It's important to protect the embryo so that it can develop properly, okay? So it doesn't die on its first day. Now, there are two other structures that the monocot has that you need to know that the dicot doesn't have. 1. Endosperm. So we notice this yellowy part we haven't labeled yet. This is the endosperm. It contains a lot of starch and proteins and stuff that the embryo will need to develop. The Dicotyledon seed gets enough of it from the Cotyledons. Okay, so they don't have this endosperm as well. Lastly, here we have aleuronone. That's this layer here. This little layer here. It contains proteins. Okay, so we know the endosperm contains a lot of starch mainly, but some proteins. The aleuron has a lot of proteins. Okay, and that's also another difference between these two. Now we know, okay, about how the seed is formed and what seed structure is. We know pollen comes in, pollen tube grows down, the DNA from the pollen, from the male gametes, travel down towards the ovule where it mixes with the female DNA to form a zygote. This is called fertilization. Then the seed grows and we know some of its structures now. Now what happens? Okay, because now we know at this point this flower has developed a lot of seeds. Okay, so the seeds are now here. Okay, now let's pretend a rose can make many types of seeds. Okay, obviously it can't, but for the sake of this explanation, we're going to pretend this rose represents all plants. Okay, so it can create this kind of seeds. It can create fruits which have seeds in them. It can create nuts which have seeds in them. Okay, so let's pretend a rose can create all of these seeds through this process that I just mentioned. Okay, now after the seeds are made, they need to be moved somewhere else, right? Why? We want them to be moved somewhere else because otherwise, say they land right here and a flower grows right here. Now these two, the mother plant, or the mother and the father plant, will have to compete for nutrients with the baby plant because they share common soil, which means they will compete for nutrients and sunlight and all these kinds of things. So it's important that we learn how these seeds are dispersed to many different places far away so that they don't have to compete with the mother plant for nutrients. So how does this happen? There's 3 important ways that you need to know. Okay, so let's pretend this it falls here and there's this nut here. So I'm going to tell you a little story that'll help you remember it. So right here we have Scrat, right? It loves nuts. Now it's trying to look for one, as you can see. It's just looking in the wrong direction. Now here we have a pod, okay, a seed pod. I don't know if you've eaten this before, peas, but it's in a little bag, and we call this bag a pod. Okay, it has all the seeds in them, you can see. But until these seeds are let out, they can't grow anywhere else. So what happens is over time, these peas will dry out so much. The seed pod will dry out so much until it dries out so much that everything just shoots out. Okay, so what will happen is it will dry out a lot and everything will just shoot out. Okay, and so these seeds will get spread everywhere. Okay, this attracts Scrat's attention. He was like, "Whoa, that was a big sound," and so now he looks the other way. Okay, and he's looking. Suddenly he spots his nut, and he grabs it, and they peace out. Okay, so he grabs it and they move. But right when he was about to grab it, this predator sees him and he starts running. And he's like, I'm gonna get him, I'm hungry. And so he runs towards him, but Scrat and his seed managed to get away. Okay, so they get away and he was not in time. But he reached now this flower. By the way, this is a humongous rose, okay? Now he comes and he was like, oh, I missed my opportunity, but I see a nice little fruit. Even though they probably eat meat, we're gonna say he's gonna eat fruit today. So he takes this fruit, he eats it, and he digests it, right? So right here, I mentioned 3 ways how seeds can be dispersed. 1, through the seed pod. They dry up a lot and they shoot it out, okay? It gets dispersed everywhere. 2, squirrels can take, or different animals, it doesn't have to be a squirrel, can take these seeds and they will take them, right, and store them for winter. So they'll bury them somewhere under the soil. So this Scrat might go and bury it somewhere under the soil for storage sakes. And then when winter comes, he forgot where he stored it, and so he can't find it. And so this seed will now germinate and become a new plant because it wasn't eaten by the squirrel after all. And lastly, animals can eat fruits because fruits have seeds in them, right? That's the annoying part about fruit. Sometimes you eat a nice peach and it has a massive seed, or you eat an apple and it has all these tiny seeds, or a watermelon, whatever. So fruits are a big way which seeds are stored in. So if an animal eats it, the seed, and it poops it out later somewhere else, 2 hours later, that was another mechanism of how the seed was spread from one place to another. Okay, so that was it for fertilization and how seed structures look like and how they are spread around. But in the next video, I'm going to talk about how these seeds, once they are stored somewhere, once they have been moved to a new place, how they become a new plant, okay? Because that's the last part that you need to know.